The Golgi apparatus is a flattened membrane-bound organelle in eukaryotic cells comprised of multiple stacks of flattened membranous discs, known as cisternae, forming a juxtanuclear ribbon structure. In simple terms, it functions as the site for post-translational modification and packaging of proteins translated by the cell. The Golgi is compartmentalized into three main sorting cisternae, the cis, medial, and trans cisternae. These make up the two main networks within the Golgi. From the endoplasmic reticulum, closest to the entry of the Golgi, lies the cis-Golgi network. Here, proteins enclosed in vesicles that have budded off the endoplasmic reticulum fuse with the outermost cis membrane and are sorted from those which need to be transported back to the ER and those which need further modification at the consecutive sorting station. The trans-Golgi network, the exit face of the Golgi, functions to sort and package proteins into the appropriate vesicles that will transport them to the plasma membrane for exocytosis and external function, or secretory vesicles which will target the proteins to intracellular destinations such as lysosomes if they were hydrolytic. Complex glycosylation of proteins destined to form glycoproteins is an example of one of the post-translational modifications that occur in the Golgi. Glycoproteins can be categorized into categories, the main ones being N-linked glycoproteins, O-linked glycoproteins, phosphoglycans, and non-enzymatic glycosylated glycoproteins. We can take N-linked glycoproteins as an example. After the initial synthesis of N-linked glycoproteins in the endoplasmic reticulum, they are transported to the Golgi and are modified by the addition or removal of sugars by glycosol transferases via the formation or cleavage of glycosidic bonds before their secretion or exocytosis from the plasma membrane or transportation to lysosomes. Other examples of modifications within the Golgi include phosphorylation of proteins by kinases within the membranes, either activating or deactivating them, and sulfation of proteins via the addition of sulfate groups. Multiple polypeptide chains can also be bound together in the Golgi to form larger multi-subunit proteins. In some circumstances, some proteins can be processed in the Golgi via self-catalytic protein splicing mechanisms which require no energy source. Here, inteins, which are highly specific sequences of amino acids, are spliced out of large precursor polypeptide chains, leaving regions known as exons present. These are then spliced together, resulting in the formation of a mature protein. The cisternal maturation model is dynamic and in basic terms depicts that the cisternae themselves are transient and have a constantly changing enzyme composition as they continuously vary from being in the cis to medial to trans cisternae state. Therefore, vesicles containing Golgi enzymes destined to modify the cargo proteins move in a retrograde direction, whilst the cargo proteins themselves remain in place within each newly forming cisternae. The vesicular transport model suggests far more static behaviour of the Golgi itself and states that the cisternae of the Golgi and the resident enzymes within them remain in place and the cargo proteins destined for enzyme activity and alteration move in an anterograde direction through the already existing cis, then medial and then trans cisternae stacks within the vesicles that bud off one membrane and fuse with the next. The rapid partitioning model is a newer model suggested, which proposes that lipids must be trafficked in the correct way within the Golgi in order to ensure that proteins are correctly trafficked through it too. The model suggests that lipids form a gradient across the Golgi, separating it into two membranous domains, one for protein processing and one for protein export. The processing domain contains glycerophospholipids, and the export domain contains cholesterol and glycosphingolipids. Cargo proteins can move between these domains and secretory vesicles can bud off the Golgi at any cisternae stack. Resident Golgi enzymes can move within the processing domain.
Within the trans-Golgi network, proteins for secretion are sorted into whether they need to be secreted continuously by constitutive secretion or intermittently by regulated secretion. Proteins secreted by constitutive secretion are sorted into transport vesicles which immediately leave the trans-Golgi network, move to fuse with the plasma membrane and release their contents by exocytosis. Proteins secreted by regulated secretion are sorted into secretory vesicles, and rather than being immediately secreted from the cell, they are stored within the cell until they receive a signal for exocytosis. The clathrin coat of the secretory vesicle is composed of subassemblies of triskelion protein complexes. Each triskelion consists of 3 legs made up of one clathrin heavy chain and one clathrin light chain. Polymerization of clathrin on the membrane induces the membrane to curve and the cargo to aggregate and bind to membrane-bound receptors, forming clathrin-coated pits. The triskeleton protein complexes form a lattice structure which is attached to the membrane by adapter proteins. Bar proteins bind at the neck to help with membrane deformation and the recruitment of actin machinery for actin polymerization. Polymerization of actin at the neck pulls the bud inwards to aid jubilation by creating a channel at the neck. Dynamin polymerizes around the neck and hydrolyzes its bound GTP to provide energy to pinch off the budding vesicle. Shortly after the vesicle forms, the clathrin coat is removed and recycled to the trans-Golgi network.